Today we have Tommy Clausner here. He is a postdoc in Oxford while living in Birmingham. And the work that I asked him to present today is about layer fMRI, which was like the fMRI part, and like the data collection was done in Nijmegen, though a long time ago, maybe during your PhD project, it seems. It is now published in eLife and under revision or kind of revised already. And there are a couple of very interesting things that I found intriguing, specifically the way of the data acquisition, which is kind of concomitantly, kind of simultaneously while having gaps in between there. But also I was intrigued by your figures. You had very creative ways of really like showing a lot in figures, and then the kind of innovative ways of kind of showing that the complex multidimensional data set that you have. So the title is Frequency and Laminar Profile of Feature-Specific Visual Activity Revealed by Interleaved EEG-fMRI. Please, Tom.
Yeah, thanks a lot for the kind introduction. The QR code that you see like in the corner actually leads to the bioRxiv version, which is now the most recent version. eLife will be updated soon, I hope, but just so you know. And with respect to the figures, one reviewer like really urged us to make them a bit simpler and like step away from the. I also really like the layout that we had before, but like one reviewer asked us to step away from the layout we had, so I now had to change the figure layout a little bit. So it's not like those polar plots anymore, which I admittedly also really liked, but let's see. Hopefully you still like the new figures as well. Okay, yeah. The project was actually quite long lasting. It started when I started my PhD and wasn't over until like 2 years after my PhD. So it like accompanies me for quite some time, this project now. And yeah, as Renzo already alluded to, it's about EEG-fMRI. And we were basically interested in visual perception. So we present the visual stimulus to our brains, to our participants' brains, and we wanted to know what's happening in there. And specifically, what's happening on the laminar level, but also what's happening in the domain of electrophysiology. And most importantly, how are they linked? So which layer are linked to which frequencies that we observe in the EEG and with respect to specific stimulus features. So it was quite an endeavor, and I just will also provide you with some background. Much of this you will probably already know, but so that we just all on the same page. If you just look on the laminar fMRI side, you probably all know this paper from Peter Kork, where he demonstrated that deep layer activity can be attributed to feedback activity. Here they presented those Canisa figures, like either an illusory configuration or a non-illusory configuration. And as you can see, there is a signal difference in deep layer activity, which hints towards feedback activity because the actual triangle that you observe is physically not there, right? So this was interpreted as feedback activity being linked to deep layer cortical activity. Then this paper you also probably know. It's just like some example that people think superficial layer activity might be related mostly to feed-forward activity. Basically, same story. We input a stimulus into our participants' brains, and superficial layers route this input from primary visual regions, for instance, to higher order visual regions via superficial layers. Just like as a relatively coarse example. From the electrophysiology perspective, we are mostly interested in alpha and gamma band activity. Here, a classic example for alpha band activity modulated by attention. So participants view stimuli either on the left or right side of the screen. And depending on where they pay attention to which side of the screen they pay attention to, the alpha in the respective hemisphere changes with contralateral alpha band power going down and ipsilateral alpha band activity going up. And the standard interpretation was, okay, alpha band activity helps to suppress unwanted information or unwanted receptive fields, basically. So that in those receptive fields where you have actually paying attention to, their alpha band activity, alpha band power basically goes down. For gamma band activity, it was traditionally being viewed as some sort of like feature binding and so, but it's more and more being related to feature processing itself. And then there's this relatively classic paper that shows that alpha band activity is most prominently observed when you present participants with grating stimuli, actually. And the higher the spatial frequency of those gratings, the higher also the gamma band power. And if you, for instance, present naturalistic stimuli to your participants, then you often observe that gamma band power is actually not as prominent as with those gratings, for instance. So this can then be maybe interpreted as some sort of either I don't know, low-level feature activity being reflected by gamma band activity, or maybe even like spiking, coordinated spiking itself. It's a bit ambivalent still what gamma is really doing, but it's mostly related to the processing of visual features, let's say, and most prominently observed in gratings or high contrast stimuli. Then there, of course, has also been some work on laminar fMRI together with EEG, like one of our colleagues, René Scheringer. He did this experiment like before and published it in 2016 already. And what he could show with this annular stimuli is that alpha band activity is mostly related to all cortical layers, more or less equally distributed. So you have this 10 hertz alpha decrease through the entire column after presenting like those stimuli, and gamma band activity being more or less limited to superficial and middle layers. But the issue there is they are having not really done a feature-specific experiment. So they presented those annular stimuli that were then either moving inwards or outwards as a task for the subjects. So you could argue that all features in V1 are activated simultaneously at once. And so there's no feature-specific signal that you can derive from this type of experiment. So this is where our experiment actually steps in, and we were asking, okay, can we differentiate this with low-level visual features? There is some work on monkeys, though. Here, some work on like figure ground separation with depth electrodes. And what Fankerkule and colleagues observed is that there is a difference between figure and ground in the alpha band, mostly in deep layers, but also still you see like the difference being like for across all the layers. Like so for figure stimuli, the activity is actually reduced as we would expect for alpha band activity throughout the entire column, but mostly in deeper layers. And for gamma band activity, it's a similar story. Gamma band activity increases for figure, and mostly in superficial layers. This is at least what they describe in the paper, but you can also see that there is some deep layer bump as well. They don't really mention this in the paper on their interpretation of the data, but as we will see, like in our results, this deep layer gamma band activity, although not being traditionally reported, might actually be important. And the shortcoming of this particular experiment is that there were no humans involved. It's monkey data, so there's still evidence lacking for this particular process in humans. So the question that we try to answer is, first of all, is gamma band activity feature specific in superficial and middle layers, as we would expect from previous experiments? And also, is alpha band power only feature unspecific? Which layers are involved, or is there maybe even like a feature-specific contribution of alpha as some theoretical models might imply? So just like to get you on the same page with our theoretical perspective, imagine you have two receptive fields in V1, and one receptive field is paid attention to, and the other is not, similar to those studies that I presented before. Then you would assume in this receptive field where you pay attention to, you have reduced alpha power and high gamma power due to the feature processing ongoing in this receptive field. And in the unattended receptive field, you would have high alpha power, basically suppressing activity in this receptive field. But you could also make a similar case for stimulus features themselves, where you be like in the same receptive field and you pay attention to a specific stimulus feature. And again, the hypothesis would be if there is such a thing as feature-specific alpha oscillations, then you should see reduced alpha power for, let's say, like features that are actively processed together with an increase in gamma band power, and features that are actively not processed or maybe even like suppressed due to the task conditions. We might see higher alpha power there, similar to what we see in receptive fields with respect to attention. There's also some evidence that this might be the case. In this specific study, what they did is they had left and right stimuli, so this typical spatial attention paradigm, but they also had color as a variable, like as a feature variable. And what you can see in the lower part of this figure is if there's no spatial cue, but the color that they reported is the important factor, then we see like this a similar um, lateralization of the alpha power if there is low feature similarity. So if the features are very different, we see like a similar alpha differentiation that we normally only observe with respect to spatial location, which is in the top part of this figure. So this is already quite promising. But of course, this is simply an EEG study and doesn't really tell us much about the layer distribution of this. But there is evidence that this could actually work. So the theoretical model, here a paper of my PhD supervisor, Mathilde Bonfond, would be we have region A, which is communicating feed-forward uh, activity from superficial middle layers to middle layers of a next region, region B, and this is communicated via higher gamma power. There's also some evidence that this might also happen in deep layers, maybe even in feedback direction, but mostly like in feed forward, not as pronounced as you would expect in superficial layers, but still could be the case. And then, of course, those two bands in superficial and deep layers in feedback direction with low alpha power reflecting the communication between those two regions. That's the theoretical model that was behind this experiment. To investigate this, we used EEG-fMRI for obvious reasons. We needed a high time resolution, but also like a high spatial resolution, of course. And the task was basically this. You would be presented with one of those stimuli, and your task would be to answer whether you see a wavy pattern in this stimulus or not. If you see a wavy pattern in an oddball, basically you would respond with a button press. If not, you would just not respond with a button press. In this case, by the way, there is a wavy pattern. So the wavy pattern is super subtle. Subjects were actually quite good at at performing this task, like after a bit of training, 95% accuracy behaviorally, I believe. But yeah, as you can see, like the pattern is like very subtle, so participants had to actually pay close attention. How would a trial look like? Coming from the previous trial, like 0.4 seconds before the onset of the new trial, there would actually be a fixation change. The red dot that you see in this fixation indicator previously was green, and 0.4 seconds after the onset of the actual trial, this fixation indicator would turn red, indicating to the subjects that they should avoid blinking now. Then there would be a 0.8 second period for recording baseline EEG data. And also from the onset of the EEG period, basically no fMRI data whatsoever is recorded. So there is like a gap in the sequence, and gradients are turned off, and we don't record any fMRI data during this EEG period of interest, let's say. After this 0.8 seconds of baseline, there would be a stimulus presentation. And this could either be a left or a right-oriented stimulus, and with a chance of like 2 times 8.3%, it could be a left or right-oriented stimulus with a wavy pattern. I exaggerated the wavy pattern a little bit for those examples there, so that you can see how it would look like in a bit in an exaggerated way. But then there's also, again, like a real-life example of how this would look like to the subjects. After this 1.6 seconds, there is the subject would respond as fast as they could during the trial. And after this 1.6 seconds, there was the feedback period where they either would receive hit or miss feedback, or false alarm feedback, or in case of a correct rejection, it would just be another pause of 0.6 seconds. And after that, 3 consecutive volumes of fMRI data were recorded. So we had always the same schedule of having 3 seconds of no fMRI data being recorded whatsoever while the trial is happening, and after the trial, 3 consecutive volumes are recorded. And this sequence of events would make up a trial. In each block, there would be 60 trials, and all things considered, there would be 4 experimental blocks. And we would record 3 volumes per trial. So like everything combined, it would be 4 times 60 times 3 volumes that would be recorded for a single participant. Before that, we have a high resolution anatomical scan of 0.8 millimeters. The actual functional data was recorded with a resolution of 0.9 millimeters, almost ISO, not perfectly ISO, but almost. Um, and after that, we also had a perception receptive field mapping with the same sequence, just without the gap. But otherwise, same parameters for this. This was mostly to determine V1, V2, V3, eventually V4. But we later settled on only using V1 data because the receptive field mapping was not as good as we would have liked. So we combined this data with some atlas data and only limit our analysis to V1. We have V1, V2, and V3, the full analysis and supplementary material, but for the main paper, we only focus on V1 because this gave us the cleanest data. Oh yeah, and we did this for 52 participants. Should have mentioned this somewhere, but didn't. Yeah, we recorded 52 participants with this protocol.
Can I ask for a second? So based
Sure.
on the echo time, it seems you used 3 Tesla. Is that right?
Yes.
Okay. And then kind of you are within each trial, you have a period where you're only acquiring EEG and then where you're only acquiring fMRI. So nicely
Yes.
kind of balanced and kind of temporally shifted because of the HRF delay.
Yeah.
Could I maybe ask, like when I read the eLife preprint, I couldn't really figure out what you mean by kind of the feature specific with respect to that task. And then, excuse my ignorance there, but could you kind of then remind me at this stage, kind of the feature, I guess, would then be the wavy pattern, right? And then people sometimes detect the wavy pattern and sometimes don't. Or how do we, like, how does this like task now relate to feature specific? How would you extract the feature specific bold versus the feature unspecific bold now? Like there's no baseline, right? You continuously only acquire at the
Yes. Yeah.
end of each trial.
I would go a bit in detail later, but just
Okay.
for now, like feature specificity would be determined by voxel responding preferably to left or right oriented gratings. So you would just select like voxel that have like a higher response to left-oriented gratings or to right-oriented gratings, or we compute like a first-level contrast between both orientations and then say like, okay, if it's positive, it's a left-oriented grating. If it's the result is negative, it's a right-oriented or a voxel preferring the right orientation. So there's those two ways we could subselect voxels based on the t-value of left or right only, or also left and right combined, just any stimulus. So this is what we would call feature unspecific, and then also the contrast between both. Yeah.
Thank you.
I will go into this a little bit more detailed just in a bit. For computing the EEG-based regressors, we first computed like a beamformer analysis of the gray matter voxel locations of the entire slab or partial volume that we recorded. The result was then virtual electrodes for each of those locations. And then we time frequency transformed all of those virtual electrodes and then selected the virtual electrode with the highest alpha response or gamma response for each hemisphere. This means we would, in the end, have 4 virtual electrodes selected, one for each hemisphere by one for each frequency band with the strongest response. You can see an example, like the average for all those selected electrodes for one hemisphere on the right side of this figure. It's a very clean EEG response. We're super happy about it. And then on the left side of this plot, you see a full brain DICS beamformer with the 5% strongest responses thresholded. We just wanted to ensure that indeed the strongest response comes from visual regions, which it does. So we were super happy about this. We did it this way because René Scherringer, in his previous paper, he did an ICA-based approach where he would select alpha and or gamma components from the ICA and then extract the data from this component and use it as the basis for the regressor. But we wanted to have a more computational approach that involves less human intervention. So we went for this beamformer approach and then just selecting the strongest decrease in a set time frequency window and then selected those virtual channels from there. What we then did is we have our time frequency resolved data for all of our channels. They are in bins of 100 milliseconds, and we would select 400 millisecond bins of this or windows of this, where we then convolve the power change with the hemodynamic response function as built into SPM, which then gives us our EG-based regressors. We z-scored the data beforehand or the power values beforehand. But basically, this is how we derive the regressors by just convolving the power changes over trial with the hemodynamic response function. For the layering, we went for a Tim van Maurik's approach, which is a bit different to how Laney does it. But the basic idea is you take your gray and white matter surfaces, or white and pile surfaces, and then you divide them into shells. And those shells are basically ignorant of the voxel locations themselves, and they take the curvature of the cortex into account. And then, as you can see, like in this example, you can take a single voxel and just like ask by how much of the volume of this voxel is divided by those shells. And then you would assign a value to this fraction that is like either in this part of the segmentation or in this part. In this case, you would say like, okay, 12% of this voxel belong to the middle layer, 80% belong to the deep layer, and 8% belong to white matter. And those values are then used to weigh the result of the GLM later. But just here as an example, like some real voxels, if you see the voxels that we selected and the layers that we could attribute them to, then the height of the bar would reflect the basically like the fraction of how much this particular voxel was attributed to the respective layers. As you can see, like it's relatively clean. But also the benefit of this is that we suspected and also tested this, you get a bit of a higher signal to noise ratio doing it like this, as opposed to assigning voxels to a specific layer and then just saying like this voxel belongs to this layer, this voxel belongs to this layer. With this, we can boost our SNR like a little bit, which is super important because especially like for gamma band activity, effects are basically tiny. So now for the feature specificity. So subjects could either see left or right oriented gratings. We would not take the oddball stimuli into account. So it would basically boil down to analyzing only left or right oriented non-oddball stimuli. And we would basically have then EEG trials, or we would have EEG data and fMRI data of both orientations, of course. And we could say we can combine trials where we recorded EEG data from the left orientation with responses of voxel that prefer one or the other orientation. This gives us kind of like this grid. So we build our regressors from left-oriented trials, our EEG regressors from left-oriented trials, and we combine them with either of those voxel preferences. So we have voxel preferring the left-oriented stimuli or voxel preferring the right-oriented stimuli, and those two EEG orientations built from the trials of those orientations. And if we then build a GLM where we combine the preferred orientations with each other or the non-preferred orientations with each other, we can say like we have a congruent or an incongruent combination of regressor and voxel preference. With this, we can then later boost our analysis by contrasting congruent and incongruent selections. Yeah, but this is how we define feature specificity in here. The GLM would then boil down to having our EEG-based regressors and having our voxel selection based on whether they prefer left or right-oriented gratings. And as mentioned before, those voxel selections were either based on the orientation alone, like how strongly does this voxel respond to left-oriented gratings, how strong does that voxel respond to right-oriented gratings, or how differently does a voxel respond to both orientations, like left minus right in this case. And for our feature unspecific analysis, it was just like how strongly does a voxel respond to any stimulus presentation. We enter this into a GLM and we get our beta value for a single voxel. And this beta value is then multiplied by our fractions that we assigned to this voxel belonging to one of the layers, like our layer weights that we obtained beforehand. We are not interested in CSF and white matter, of course, so we only report results for superficial, middle, and deep layers. And after averaging across trials and blocks and so forth, we end up with a result matrix that is depth by frequency, with depth being those three layers and frequency being whatever frequency band we've just selected. And of course, we have this type of matrix for every participant. What we then can do is run a cluster permutation test on this depth by frequency matrix, and we get, let's say, like a significant result there, which then tells us, okay, there is a significantly, let's say, like alpha decrease in this frequency range and is distributed across layers like this. What we still don't know yet is how strongly each layer responds actually. And this is quite an issue because when you just say like, okay, we take the average of every layer and then compare those, you run into issues with respect to multi-comparison. You have a lot of like statistical power issues because the effects are so tiny. So what we did is we came up with a little bit of a creative way on doing the statistics on the layers themselves. And this was like also an in-house developed statistical test for this purpose, which we call the AROS test, which writes out to autoregressive rank order similarity test. I will just quickly explain what this is. The first step would be you average your response across the part of your results that span the widest frequencies, right? So you would always average along the frequency dimension, but with the widest possible window, and then you get an average profile of something like this. You compute the rank order of those averages, and then you have like the strongest response, the medium strongest response, and the weakest response. What you then can do is you can actually fit your data to this rank order, and this gives you some value. So how well does the data actually explain this rank order? You then shuffle the data and recompute this procedure. So you shuffle the layer labels basically, compute this average rank order again, and then just check again how well does now everything fit to this random rank order basically. And you do this a lot of times so that you build up a permutation distribution, and later on compare your initial value to this distribution. What this tells you then is how strongly or how likely is it that your obtained layer profile is actually meaningful. You cannot make any statements anymore on how much stronger, let's say, like superficial layer is compared to deep layers, but you can say that this rank order is actually meaningful, that superficial layer is stronger than deep or middle layer, just not, you cannot tell anymore by how much. So you sacrifice statistical power on the difference itself or meaning, if you wish, but you gain statistical power on the rank order itself. Yes, so the final result structure kind of looks like this. We have our superficial, middle, and deep layers from our laminar fMRI. We have feature-specific results and feature-unspecific results. Remember the feature-unspecific results being those that where voxels were selected based on a response to any stimulus. And we have this for alpha and gamma. For the feature-specific part, we can separate them into congruent and incongruent subselections, with voxels preferably responding to left-oriented gratings and EEG regressors built from left-oriented trials, forming a congruent pairing, and vice versa for incongruently selected or incongruent GLM models. And what you then get in the end is something like this. So this would be the results for our feature-unspecific analysis. The top is all positive responses to any stimulus, and the bottom would be a negative bold as a response to any stimulus. So we have those two voxel subselections, and we enter them into a model with the EEG regressors built also from any stimulus. And what we can see here is that alpha and the bold response is inversely related. So basically, if alpha goes down, bold goes up, and if alpha goes up, bold goes down. For positive bold responses, we see that there is no layer specificity whatsoever. And for negative bold responses, we see that we have an increased effect in superficial layers. So again, we cannot tell by how much stronger this superficial layer is as compared to middle or deep layers, but we see that the rank order is superficial, middle, deep in this case. So we see, okay, the strongest effect for negative bold related to alpha is in superficial layers. In the gamma band, we haven't observed any effects for this specific analysis. At first, this was a little bit surprising because we would have expected a feature-unspecific gamma band response just based on the idea that, okay, there are stimuli on the screen, like gratings on the screen. So we should see a relationship between gamma and the Bolt signal. And we also found like a really strong gamma band response in general. So why is there no relationship with the Bolt signal here? I will come back to this later, but just so keep in mind, like for the feature unspecific response, we don't see any gamma band effects. What we also did is we wanted to see how different alpha subbands actually respond to this task. So we averaged across layers and just looked at lower alpha bands, which is 8 to 10 hertz, and upper alpha subbands, which is 11 to 13 hertz. They were also like determined based on the average gamma band response from the EEG. And we have a significant interaction there for Bolt signal direction and upper or lower alpha. And the simple effects tend to point towards an effect like mostly like in a positive regime where it's not significant. It's just like trend level, almost 0.1, but it hints towards mostly upper alpha being responsible for this effect there. But we don't know. The interaction is significant, but there's only like a trend level effect for the simple effects there. Now let's have a look at the feature specific results. Those look super complex, so I break them a little bit down. The first part would be, again, we see alpha decrease, Bolt increase. So we have an inverse relationship between alpha and the Bolt signal. And if we look specifically at congruent combinations, then we see this effect is mostly visible in superficial layers. So congruently combined EEG regressors and voxel selections mean we have this inverse alpha effect. Alpha goes down, Bolt goes up, mostly in superficial layers. We observe the same for incongruent combinations, almost significant. I tend to count it because it's 0.053, so it's debatable. It's also FDR corrected because we use different voxel selection thresholds. So uncorrected, it would be significant, but FDR corrected, it's not anymore. But again, assuming this would be an effect, then we would see this as well in superficial layers mostly. However, if we contrast both congruent and incongruent selections, we see that the effect is now in superficial and deep layers. What does this mean? This means that for the congruent combinations of voxel preference and EEG-based regressors, we see that the decrease in the deeper layers is stronger in the congruent than in the incongruent bit. And if we then build up the contrast, we see it there. What we also can already see just by naked eye that there might be a difference in the alpha subbands. So incongruent selections tend to be like more on the lower end of the spectrum of the alpha, while the contrast between congruent and incongruent tends to be biased towards the upper part of the alpha spectrum. And indeed, if we compute a mixed effect model on this, we see that there is like a significant interaction, which seems to be mostly driven by upper alpha being lower or having a stronger effect for congruent and a weaker effect for incongruent voxel selections. This means this feature-specific response seems to be mostly driven by the upper alpha range. And going back to the contrast in the congruent minus incongruent in the layer results, being mostly in superficial and deep layers. I will go for I will explain like how we interpret these results a bit later, but just so that you have seen it there. And again, no gamma band effect, which was super disappointing, to be honest. But then we thought, okay, those voxels that we used in this specific analysis were based on the contrast between left and right oriented gratings. So we did a first level analysis, left minus right, and then took like those that responded strong, like where the result was the most positive or the most negative, which then would point to voxel preferring the left or the right orientation. This also means it probably includes voxels that respond like more or less positive to the respective stimuli, but could also include voxel that in itself respond negative to specific voxel because we didn't make a subselection there. So in the next step, we did the same analysis, select our voxel based on the contrast between both orientations, but then limit this subselection to voxels that strictly respond positive to either stimulus orientation. And what we see here is the alpha effect gets weaker. So, which probably means that not making this subselection, but not excluding negatively responding voxels, actually helps the alpha effect there. So This again brings us back to our feature unspecific results from the beginning. Alpha is related to bold decreases seemingly, which is also reflected in this feature specific contrast here. We see a similar-ish pattern to what we have seen before, like with these feature-specific results. Now the effect being mostly limited to congruent voxel subselections, but again, the responsible driver of this effect seems to be the upper alpha band. And voila, we now have also a gamma band effect. So our interpretation here is, first of all, gamma band activity is limited to positive bold responses, which makes sense because in this previous paper that I showed you by René Sreringa, he also limited his analysis to positive only bold signal. So we find this as well. But we find it only for the contrast between congruent and incongruent, which means the effect is probably super, super weak. So for congruent only, we see there is some sort of positive response. For incongruent, we see even some sort of tending towards being a bit negative response, and only the contrast itself actually is significant here. So it's a super tiny effect, but it's basically what we would have expected, that gamma response features specifically here. We have observed no layer specificity, which also puzzled us a little bit. But when you then change the threshold for voxel selection a little bit, in this case, we use the 10% most activated voxels for our GLM. And if you lower the threshold a little bit to the 25% most activated voxels, then you see actually a layer specific effect for gamma, probably because signal to noise ratio increased a little bit, and we see superficial and deep layer effects as we would probably have expected, also based on the monkey data there, which is super cool. Okay, in summary, what did we find? For feature unspecific bold EEG relationship, we found that a bold increase leads to a alpha power decrease, and a bold decrease leads to an alpha power increase, or vice versa. We actually don't know the directionality really. And this is mostly irrespective of upper or lower alpha band subselections. I put this lower alpha there in brackets because it seems that the effect is mostly driven by upper alpha, but it's not really certain at this point for the feature unspecific analysis. And we see that the negative bold is mostly related to alpha in superficial layers. For the feature specific results, we see that the change in the bold signal is inversely related to alpha, mostly in the upper subband and mostly in deep and superficial layers, or superficial and deep layers. We find no gamma response for feature unspecific analyses, and we do find a positive relationship between gamma and the bold signal for, again, superficial and deep layers, kind of as expected. How do we interpret those results? Well, we think when you feed in a stimulus to a column in V1, to middle layers mostly, then this information is processed and fed forward via mostly superficial layers. And gamma thereby reflects the activation of a feature selective column. So you have a column responding preferably to left orientation, and this column processing actually the left-oriented stimulus is accompanied by an increase in gamma band power for this specific column that prefers an orientation. For alpha, we think there's maybe two alphas there at play. There is some literature recently pointing towards probably alpha not being a unified process, but probably reflecting different processes in a similar frequency range. And our idea here is there might be some sort of globally modulated alpha, which could reflect attentional processes or simply organizational processes, routing processes. There's many things that you could imagine, but there's this more globally modulated alpha, which is more on the lower end of the alpha spectrum and reflected in superficial layers. And there is a more feature-specific alpha component that is probably related to deep layer activity mostly. And this would then disinhibit feature selective columns, while the globally modulated alpha would more or less be responsible for disinhibiting active receptive fields, if you wish. Yeah, and with this, I would like to end and thank you for your attention, and especially thanks to Mathilde Bonfond and René Sreringa, who accompanied me for this very long journey. and I'd be very happy to take questions and be looking really forward to eventual discussions.
Thanks a lot, Tommy. Wow, that was quite a bit, and then I'm still needed to wrap my head around all these regressors and this quite deep and comprehensive analysis of looking at differences and differences of differences with all kind of different dimensions that you have. It's quite multidimensional.
Yeah.
Maybe I can start with maybe two questions. The the first one was about veins. So you know that we have the pile veins and the straining vein effect, and I guess the strongest response that you're seeing of this anti-correlation between the oscillations and bold, it's dominated from the superficial layers in alpha, and that could mean that this is mostly driven by veins. And then, for example, for the other results where you have this upper deeper, I was wondering whether you could comment on this spatial deconvolution approach from Tim Morik, because in my experience, when I tried something similar, I did very often kind of, it's like a sharpness filter, right? So anything that kind of depending on your slope, it tries to make anything like any spatial feature and amplifies it. So that when you had a slope, it can make it like a U shape. Maybe the second question I would have was about the spatial selection of your virtual electrodes. Like here in the introduction, you talked about kind of attention and spatial specific features. So in the EEG results, the virtual electrode looked like they're not only containing V1 or like not only the screen size of V1. So I was wondering if you could comment on kind of the effect of V2 or spatial vignetting or those kind of things and whether they matter for this kind of feature selection of left and right.
Yeah, thanks. For the first question with respect to the or laminar profile, let's say. For this analysis, I think we are pretty safe because we compute a contrast between congruent and incongruent. And if there would be any differences in signal to noise ratio, they would affect congruent equally to incongruent model combinations. So if we then compute the contrast between congruent and incongruent, it should probably account for this. On top, we are not really measuring bolt activity per se, right? So we only measure the relationship between power changes and bolt signal changes, which also helps a little bit to make this veinal draining effect probably not being as important. But we also computed like a TSNR analysis on this. And what we found, ironically, is that we have our highest TSNR in deep layers, and then middle, and then superficial. I was a bit puzzled about this, so we also computed the layering based on your layering software on Laney, which you see like on the right, but we found the same. Depends a little bit on what type of SNR measure you use, but the tendency is that the SNR or TSNR profile is different from our layer profile results. So this is a bit reassuring. But to be blunt, we didn't take any veinal draining effects into account. We didn't model them at all, mostly because we were interested in how power changes relate to bolt signal changes, and both were Z-scored. So in this case, I think it's less important because we are not interested in the absolute signal change in bolt per se, but only in the change related to the power change. Does that make sense?
Sure. Yes, the like TSNR, I guess you're using a slab selective 3D API as they like in Nijmegen, so which would explain that you're looking at a T1 effect here, that basically you have more saturation at the superficial layers, so the TSNR is lower there compared to the deepest layers, which makes sense. I was referring more to the gain, like that the baseline blood volume in the superficial layers is bigger, so kind of even though you have lower TSNR, the response magnitude might change, and therefore the difference between the response magnitude as a multiplicative kind of gain might affect this. I'm not sure if this would affect your kind of rank ordering though, because I don't understand it well.
I totally agree. We didn't take this into account, mostly because we felt pretty sure about this contrast in here, because this effect that you're describing should affect congruent and incongruent voxel selections alike, right? Because they are the same voxels, just paired with different regressors, if you want. So this effect should be in there in both cases. And if you then do the contrast between those, this effect should be contrasted out, right?
Right.
And this would be the congruent minus incongruent selection there.
And this is all in that 10% most significant voxels, right?
Yes, we also have it in the supplementary material for the top 5% and top 25%. But yeah, so but this particular are the results for the top 10%. Yeah. And for the other question with respect to virtual electrode selection, oop, where do we have it? There, yeah. Here the thing is, it's relatively unlikely, especially like with EEG, that you will do a source reconstruction and be able to differentiate between V1, V2, V3. So from a spatial resolution perspective, you might potentially be able to differentiate between both hemispheres in the visual cortex. So this is why we just opted for both hemispheres. But you're right, it's not guaranteed that this activity only stems from V1 or only stems from V2 or V3, or it's probably a mixture of all of them. But when we look at the results for V2 and V3, they are comparable, let's say, not identical, but comparable. So given the stimuli that we used and given how, let's say, like spatially smooth or smeared the EEG signal is anyway, we think is as good as it gets with EEG there. Although I would probably not claim that the EEG regressors stem from V1 per se. I would say that they correlate with V1 the way it does, like with respect to the Bolt signal, but I would not say that the EEG signal is a pure V1 EEG signal. Yes.
Would it matter for your conclusions?
No, not really.
No.
But yeah. I mean, at first, like at the very first version of this analysis, we indeed like had the idea of modeling V1, V2, V3 like for EEG as well, because we could just select from fMRI V1, V2, V3, right, and then just compute the virtual electrodes there. We had an extensive discussion with Robert Osenfeld about this topic, and he was like, are you assuming you can functionally differentiate those regions in EEG? We were like, no. And then he was like, why are you trying to differentiate? And then we were like, yeah, okay, makes quite sense. So we then settled for hemispheres because there's research showing that you can, in fact, separate them across hemispheres. And yeah. I would even assume that if you only use one regressor that combines both hemispheres, it would probably provide even similar results. But yeah,
Are there questions from the group? Please.
Yeah, hi. This is so interesting. My questions at this hour are never well formulated because I'm in California, but I was just thinking about everything's voxel level. You're never using any kind of contiguous clusters of voxels. And I suppose that's because you're characterizing each voxel in terms of the proportion that's across the different layers. Is that right? So it wouldn't work if you're doing clusters, right?
Yeah.
Okay. Thank you.
I mean, the idea is it was quite interesting when we did the TSNR estimate. I played around a little bit with the data. And what we also tried is making like a surrogate Bolt signal, like combining multiple voxels and basically constructing a signal weighted by those layer weights. But you then end up indeed getting like less signal to noise ratio as compared to the way we did it now. So I think like even though this was not really the intention was just like simplicity. But even though if you would do it or try to combine them into a single cluster of response, you would still end up with less SNR than you do with this approach. So yeah.
Are there any other questions? Maybe
By the
I
way,
have understanding question. The congruent, incongruent, that slides went fast, and then maybe I didn't fully understand. How did you define it? They're the same voxels, are they? It's
Yeah. I mean, you could say I have a set of voxels that respond preferably to the left orientation.
Mhm.
And then I have two types of, or actually three types of EEG regressors. I can build EEG regressors from power changes of the response to any stimulus. I can build my EEG regressors from power changes of stimuli that only left stimuli or only right stimuli. So I have both left and right. Like I have trials for those three sets, basically. Then I can build my regressors from those three sets and enter them into a GLM with the voxels that prefer the left orientation.
based on the EEG.
No, based on the fMRI. Like I do a first level contrast. I like which voxel prefers or like responds strongest to left-oriented gratings, let's say. And then if I then compute my final analysis with a GLM with a EEG-based regressor computed from only the left-oriented trials, then I have a congruent comparison. And if I do it for all the other trials, like that are right-oriented, then I have like an incongruent comparison. Like I measure how much the power derived from right-oriented trial, or like the power changes from right-oriented gratings relates to the activity in voxel that prefer the left orientation. This would be an incongruent comparison.
Gotcha.
So in the end, since you combine like left with left and right with right, or left with right and right with left, you end up having always like the same amount of regressors and the same voxels, basically, just combined like one way or the other
Mm.
in the final model. So this contrast between congruent and incongruent there is then basically a boost of the results in itself.
Gotcha. I think you never have any rest, right? Or is it possible to, or does it make sense to kind of um, make this more intuitive for me by comparing kind of left versus rest and right versus rest? Or it wouldn't add anything, right? Yeah.
We also had a reviewer asking this question, and I think in an ideal world, it would be nice to have rest.
Mhm.
But given that the subject was already in for almost 3 hours, including like EG and everything, preparations, it's just not really feasible. It would also be nice to have another condition where we actually manipulate attention. It would also have been nice, but then it was just not feasible, like to include like everything we would have liked. So yeah.
You didn't measure attention, did you? Like with eye tracking or
No.
something? No. No.
What we have though is a bit of an interesting finding. I have this in my bonus slide somewhere. No, where is it? Ah, there. Um, so if you look at the very right of this is like a plot from the paper. If you look at the very right, you see the t-value distribution for single orientation or the contrast between both orientations. And why can I move my... Ah, yeah. And if you look at the t-value distribution for single orientation gratings, then you see like at the calcarine, it's basically like divided into decreases and increases in a way, which is a bit strange, or we wondered about this like a little bit before. Then we looked a bit into the literature, and it seems that this is an attention-related pattern. So in order to solve the task, you don't need to watch at the entire stimulus, right? You can just focus on a small section, and that's it. And when you then look into what participants actually prefer to look at stimuli in general, like with eye tracking studies and this kind of things, and then relate this to fMRI studies tracing or trying to trace the relationship between spatial attention and bold activation, then you might be tempted to assume that this pattern is actually a spatial attention pattern. But we don't know really. But yeah.
So
But
the
our
difference between top and bottom is now the way how you contrast
yes, yeah.
left versus right. So
Yeah.
the top is a division, the bottom is a subtraction. Is that
Oh, no,
right?
the top is an OR and the
Okay.
bottom is a subtraction.
Do you think there could be vignetting in this as well? Because
No, to be honest.
it is kind
I
of
mean,
unidirectional.
what
Yeah.
I, what ensured us a little bit at least was that the contrast is a very nice pizza plot. So we don't have any spatial patterns in the contrast itself, which is good. It was a bit of a sanity check. But this clear cut at the calcarine at the top is also something we don't really know how to explain like thoroughly. So
All right. If there are no further questions, I thank you, Tommy. And for everybody, there will not be a meeting in 2 weeks, and there will be a summer break, but we will meet again in 4 weeks with Saskia Bollmann talking about setting up layer fMRI on the impulse scanner. Right. Thanks.
thank you very much.